My challenge today is to talk and focus on stewardship. So we've talked a lot today already about the importance of antimicrobial stewardship. Here we go. I want to pick up on some themes around biomarkers. There's been a really big effort from UK government through the Department of Health, now the Department of Health and Social Care, and the NIHR, which is its arm that funds and delivers research in the UK, to try and progress over the last 10 to 15 years evidence around the use of biomarkers. There have been a lot of questions today about biomarkers in routine care, so hopefully we can address some of those over the next 25 minutes. Can I just ask a question? Who in the audience has helped one of their patients take part in research? Okay, so that's a minority. So it is a national ambition for every patient that we have contact with to invite them to take part in research. You are a really critical part of advocacy for sepsis. All of you in the room, everything you're doing. So one of the probably the most important learning objectives of today, when you get away, think about how can you enable your patients to take part in really important national research. So that's for you to think about. My objectives today are to focus on antimicrobial stewardship, to think about our current best practice of the use of biomarkers in antimicrobial stewardship. But I'm going to lean into international guidance and then talk a little bit about NICE's response, because obviously we work in the UK. I'm going to tell you some new results and some emerging results that might help you in your current practice. And then, of course, because a lot of my salary is funded by the NIHR, I've got to persuade you that it's really important to take part in research. And I'm going to name some studies you might think about. Okay, so I'm not going to dwell too long on stewardship. We know it's critically important that we've got to look after. We are the stewards of these really important and potent drugs that are going to save life around the world. But you know, particularly in sepsis care, this is a real challenge because we've got to do our best to look after those drugs, to give them to the people that need them most in a timely fashion, in a sufficient spectrum to hopefully tackle the bugs that they're like to be infected by at an appropriate dose, and then end them at an appropriate time so the duration is appropriate for that care. Really big challenge there. And you know all about that. So stewardship is critically important because, as you know, this is an existential threat to the human race. If we don't get this right, healthcare in the future is going to be impossible to provide. Basic oncology care, basic elective surgery is critically going to be threatened by this. And this will lead to all sorts of sustainability issues that are well documented. So that's my soapbox bit. So when I'm on the ward, on the intensive care at my hospital at Salford Royal, part of the Northern Care Alliance, I think of three things when it comes to antimicrobial stewardship. So the actionable things around making decisions to start an antibiotic, when to review and think about whether that choice was correct, and then when to end my antibiotic. So three critically important actions that are going to impact on overall stewardship. So what does the international guidance say about the best practice? Well, firstly, there's pretty good evidence that we should see our patients every day. So I think that's fairly non-contested, really. I should go and see I should turn up for work, shouldn't I? I mean, that's what I'm paid to do. So I should go and see and review my patient every day with a multidisciplinary team. And you're all part of that multidisciplinary team. And if I forget to think about sepsis, forget to think about the use of antibiotics, you, for goodness sake, remind me. You know, we should challenge each other around best care. Okay. There is pretty good evidence that we should have good surgical source control. It's pointless flooding patients with broad-spectrum antibiotics if we haven't thought about a surgical cause that could be removed, or a line or a device that could be safely removed in that patient that may get rid of the source of infection. Critically important. And finally, we're told to aim for shorter antimicrobial courses. We think nationally this is a good thing, but we haven't got much advice about how to do that. And I can remember some years ago, you remember this when Sally Davis was the Chief Medical Officer, we were asked to reduce meropenem use in the hospital by a fixed percentage. Nobody actually told us on which patients, but that's what we had to do. And we were incentivized to do that. So the notion of decision support in this space is critically important. Okay, so about the short duration. So there is further international guidance around duration, and it's very interesting that in most observational studies, that the use of a fixed duration approach, so I am going to prescribe for a week, or I am going to prescribe for 5 days, is less effective and more harmful to patients than if I go and review that decision on a daily basis. Okay, so fixed duration can be problematic, and we're not recommended to do it. And I suggest when you go back to your current NHS practice, you will find a lot of fixed duration prescribing going on in a very stressed system where we make a decision and then don't revise that decision for a period of time. So fixed duration, not recommended. Okay, so what are we doing about this? So we're doing a study to look at this very question. There have been no randomized controlled trials, I think in most jurisdictions in the world, to ask the question, is short fixed duration prescribing, and in this case, 5 days for sepsis, against routine care, whatever that is in your hands, in a randomized fashion. We are doing that study now. This is led by Tom Hellier in Newcastle. If you're from that part of the world, you'll probably know Tom, and I'm supporting him in that in terms of this study that is funded by the NIHR. We've just got through pilot, so we've recruited the first 20% of patient cohort. It's requiring 2,000 patients to be certain that we can make a difference here if a difference exists. And that will be reporting probably over the next couple of years. If you want to take part in that study, there's an email address there to get involved. Really important study. But now I want to go on to markers, because that doesn't involve biomarkers. So Ron's already said that when he and me both think of markers, we think of infection indirect markers, so inflammatory markers, CRP, procalcitonin. I was going to do a piece of asking you what your favorite markers were, but perhaps time won't allow me. You can think about which of your famous favorite inflammatory markers, and we've heard about another one today that joins that group of important decision support markers. And then secondly, direct pathogen detection. I'm going to say a little bit about that. That's a murky area that is evolving, and we're trying to get some systematic evidence in the NHS, and indeed increasingly with a particularly new way to do this with the Health Protection Agency around rollout of a technology. So let's look at indirect infection markers to make decisions about starting antibiotics, so initiation. So what is the current international guidance? Don't do it. And why do they say that? Why does the sepsis survival campaign say that? Is that the evidence doesn't support it. But actually the evidence is pretty low quality and doesn't always involve sepsis patients. I've got a paper there from a few years ago, which is actually in the setting of community-acquired pneumonia, of which a percentage of the patients had sepsis. So this was a very nice randomized controlled trial done by Derek Angus's group in Pittsburgh in the US. Really big, well-organized study in the hospitals that they run in Pittsburgh. And they had decision support in the ED for community-acquired pneumonia. And they used PCT point-of-care measurement and a protocol such that was randomized between patients. Some patients got usual care, some patients got the addition of procalcitonin. What was really interesting is that the emergency physicians followed the advice of PCT, but it didn't make a longer term impact on the use of antibiotics. So in the hands of the emergency physicians, it worked pretty well, but then half the patients went back to the community, half went to the floor, some to ICU. Those decisions were upended, and antibiotics were prescribed either by primary care physicians or by hospital critical care physicians. And you can see the results here, that there is no difference in antibiotic use between those two groups. Really important evidence, but it doesn't completely tell us the full story. So I'm going to come back briefly to a study that is going to report soon in the NHS on that question. So that's critically important for you to look at. Now, the next question is, can we use these inflammatory markers to stop antibiotics? Can they help decision support some days later to say, well, actually, this patient's had enough? And the advice, interestingly, the current international advice is maybe it works in that setting, but the evidence isn't strong. Okay, so it's weak advice, and it particularly refers to procalcitonin. So the daily measurement of procalcitonin and looking at patterns and looking how it falls may actually be beneficial to reducing antibiotics in the NHS, but we don't know that yet. And this study, which was the SAP study, some of you may know, it's probably the best one that's been done to date. It was conducted, a multicentric study in Holland, reporting just before pandemic, and showed that you could reduce antibiotic duration using daily procalcitonin in critical care, some sepsis, some not, by about 2 days. But strikingly, in doing that, you could reduce mortality. And that feels remarkable, doesn't it? Mortality in critical care reduced by 10%, linked to a difference in antibiotic use. Now, there's a problem with this study. It's an open-labeled study. So the biomarkers were delivered to half of the patients on a daily basis, and the control group didn't have that intervention. That was wild care, as you might call it, or usual care. So it means that the multidisciplinary team on a daily basis were asked to go and review the patient and review everything about that patient to make a decision to stop antibiotics. So there might have been something else going on in the trial, what we call performance bias. So there might have been other care going on, not just the biomarker. So that's probably the best evidence we've got up to this point, not actually from the NHS, and a sort of cautious, well, it might work, why not try it, but make sure you're reviewing the patient daily. So NICE had a look at this just before, well, 2015, remarkably, so it's quite a long time ago, and they came to the same conclusion, might be useful, but we don't really know. We can't really recommend the use of PCT in this setting. There's a real lack of UK-based evidence, and so there was a recommendation to do some high-quality research. So I'm going to present now a couple of studies that have tried to help address these objectives. You can see here that they're also interested in CRP. The evidence around daily CRP is not quite as good as PCT, but we measure it, we've got access to it. So what does that mean in the system? So, so the Pronto study is currently in peer review. It's going to be reported over the next few months. It's led from colleagues in Liverpool, and they were looking at, does procalcitonin in combination with the news score improve your ability to make decisions about antibiotic prescription? Does that continue into the hospital or the community? And is it safe in terms of mortality? So that's the first study we've ever done in the UK, really important study. You might not be able to see this from the back, so I apologize. There is a link in this slide to the protocol. But basically, these patients were randomized between two groups, where the news score is how you currently use it. So there's a kind of high risk, really concerned about sepsis, and then lower risk, we might have a bit more time to make decisions. But the intervention was about getting senior healthcare professionals to review the patient quickly. And in the intervention group, that was changed by the addition of procalcitonin measurements. So we're really interested in, I know the results, but we can't talk about them yet, but because they're in peer review. That's finished, November 23. It's taken a long time to put the data together. A lot of engagement. It may even been that your hospital contributed patients to this. So look out for this paper. It could be game-changing in your setting. So this is the trial that I led in critical care nationally, which has now completed and is reported. And the link to the paper is in JAMA from 6 weeks ago. So we were asked by the NIHR and NICE to take on the challenge of daily PCT measurements or daily CRP measurements against standard care to make decisions about stopping antibiotics in critical care. So the population was critically ill adults. It was about antibiotic discontinuation protocols, as I say, with either CRP or PCT. Those were quite complicated. You can read about them in the paper, but the decision support was given as advice, not the biomarkers. So we concealed the biomarkers. We didn't say whether it was PCT, whether it was CRP, or whether the patient was in standard care. Everyone got a blood test every day. You didn't know whether you were in the control group or not, and the advice came back to teams every day. Pretty risky study to do. We thought probably we would have a negative trial, that these markers would not make an effect in a blinded study. It's completed, lots of centres, 41 centres around the country, recruited 2,600 patients. And hey presto, if you haven't seen this paper already, I was quite surprised that PCT worked. It produced a meaningful difference in antimicrobial prescription right over 28 days. So not just for the antibiotic sepsis period, it actually worked for that period, but also this signal was there for 28 days. So an important signal. You might say, well, 10%, that's not much. But remember, we're trying to make incremental differences here with antibiotics that are, that we need to preserve. So we'd have met the objectives of the last CMO with this study using procalcitonin to reduce by 10%. We didn't find that with CRP, and there was actually potentially a signal for harm for CRP, and you can look at the paper. Where are we up to with this at the moment? Well, we now want to bring all the data together. So we're meta-analysing all the data with the previous open label studies, because NICE want that. And we've completed the health economic analysis, I can tell you that. It hasn't been peer-reviewed yet, but based on the meta-analysed data from the world literature, it looks cost-effective. So we've got a QALY, so it costs about £2,000 for every one quality-adjusted year gain. So it's not often you see in critical care that level of opportunity. So that's coming soon when it's peer-reviewed. There will be an update of national guidance. I have no idea what NICE will say. I'm not part of that, but that will come. We are doing some trial process evaluation to work out which bits of the trial we can then move into NHS care. And rather than just move it into NHS care, we want to do an implementation study, a quality improvement study that makes sure that we can at least get the same results as the blinded study that we did. We need now to put procalcitonin into open care and work out that we achieve that. If you're interested in taking part in that, there is a QR code there to fill in a survey that's currently live, because we're in the process of designing that implementation study with the NIHR. And please, I hope you'll be able to join in and answer some of those questions. What about children? So I've talked about, my study was about adults. So this is Enitan Carroll's study from the Alder Hey, a multi-centre childhood sepsis study, an infection study looking at the use of procalcitonin. And this was in a group of children throughout the ward, so not just in intensive care, at risk of infection and sepsis. Nice study around all the big pediatric centres around the country. It was called the Batch Trial. It's recently been published in Lancet Family of Journals. And unfortunately, Enitan didn't find that the same results as us. So in children, the PCT didn't produce the savings in antibiotics that we saw in adults. And one of the reasons for that, I think, is that the children didn't have long antibiotic courses, particularly in the ward. So the opportunity to benefit from daily PCT measurements wasn't really there. So at least in the population she looked at, it didn't have benefit. It doesn't mean PCT doesn't work. And indeed, there you go. That's the antibiotic prescription in the two groups. The average number of days of antibiotics in the trial was only 4 days, so quite short courses actually. So the pediatric doctors are doing a great job already. I've got 5 minutes, so let me just finish on something around direct infection biomarkers. So we've been interested in this for 15, 20 years, and it's a real challenge, this. So you will be familiar that we have to do blood culture, and I've showed you 2 sets of culture there. So another learning objective is to go away and find out in a sepsis patient why you should do 2 sets of culture and not 1. That is a British standard. Okay, if you're not doing them, go and find out why. But blood cultures are very important to us, aren't they? So when we get information back from blood cultures, that can be really important, but often they're negative. So the diagnostic accuracy of blood cultures is not great. We don't believe a negative result, but we believe a positive. So we have technologies as well in a positive culture where we can look at a molecular level really quickly what those bacteria or what those pathogens are, and something about the likelihood of resistance to certain antibiotics. So not the whole piece of information, but important information. So some nice technologies there, but what we're really interested in is, can we do this straight out of blood? So can you take a small EDTA sample of a few mils of blood and really rapidly find a pathogen signature at a molecular level in that? And this was a review of the available instrumentation to the NHS back in 2016 that we wrote internally for the NIHR and for NICE, based on some big studies, observational studies we'd done with these technologies. And the answer is they look really promising, but there is, let me go on to the next slide. But so there was some nice guidance on this. They look really promising, but what we need is really good clinical trials to test them for actionable questions, okay? So we need to determine the clinical scenarios, and one of those might be in the setting of invasive fungal disease. We don't often see this, but we often prescribe antifungal drugs. So the opportunity to intervene in that setting is quite amazing. If you've got a good test where negative you believe, and you could stop the antifungal drugs, that's an actionable opportunity. So what about assessing together with other markers, so pathogens with host response, adults and children, and for goodness sake, do it in routine NHS care. So I'm going to talk about very briefly in the last 30 seconds 2 studies that are ongoing that you might like to take part in. The A-STOP study is looking at a whole array of fungal rapid tests straight out of blood that could be used to rule out invasive fungal infections. So that's halfway through. You're going to see very soon a peer-reviewed paper that tells you how to exclude invasive fungal sepsis with that. So the second part of this study, A-STOP2, is about to launch, where we're using that package in a randomized way in an intervention study in patients on antifungal drugs to rule out invasive fungal disease and stop the antifungals. So that's A-STOP2, will be coming to your centre soon. It was quite good engagement with the diagnostic study. And finally, we have a platform trial led by Tony Gordon at Imperial alongside a number of us, looking at bacterial sepsis. This is underway. We've got about the first 5% of patients recruited at the moment, and I'm leading the diagnostic part of the platform, looking at the use of a rapid bacterial straight out of blood technology that's available to the NHS in association with PCT, and how can we stop antibiotics early in that group. So we're really trying hard in this space. And in summary, because you're going to say, well, what am I going to take away from this? Firstly, offer your patients research. Secondly, there's some things that work and some things that don't. And finally, there's some trials you could get involved in, including a future implementation trial of PCT. I better stop there, Diane. Thank you.